Welcome to STEM Talk.
STEM Talk. STEM Talk. STEM Talk. STEM Talk.
STEM Talk.
Welcome to STEM Talk, where we introduce you to fascinating people who passionately inhabit the scientific and technical frontiers of our society.
Hello, I'm your host, Ken Ford, IHMC's founder and CEO Emeritus. And rumor has it that I also chair the double secret selection committee that selects all of the guests who appear on STEM Talk. And by the way, all votes are unanimous. Today, we have a real treat for our listeners. Longtime STEM Talk fans know that we've done several episodes that touched on regenerative medicine. It's an exciting field that researchers and clinicians around the world are embracing for its potential to tackle some of the toughest medical conditions. But as with much promising medical science, the current conversation around stem cells is a mix of hope, hype, and more than a little disinformation. Joining us today is Dr. Mari Dezawa, a leader in stem cell research and regenerative medicine more broadly. She is known for her discovery of Muse cells. Muse is short for Multilineage Differentiating Stress-Enduring Cells. Mari is a professor and chair of the Department of Stem Cell Biology and Histology at Tohoku University Graduate School of Medicine. She has published more than 200 scientific papers and in 2018 was named a fellow of the National Academy of Inventors here in the United States. She has also received numerous honors, including the Japanese government's prize for the commendation of science and technology. Before we jump into our conversation with Mari, we have a little bit of housekeeping. First, a big thank you to everyone who has subscribed to STEM Talk, and an extra thanks to all of you who have left those wonderful 5-star reviews. If you're new to the show and you like what you hear, please consider subscribing. And if you enjoy today's episode, we'd love for you to take a moment to rate the show and maybe even leave us a review. It really helps us reach more people who might enjoy STEM Talk. As always, the Double Secret Selection Committee has been hard at work, scouring iTunes and other podcast apps for the sharpest, funniest, and most over-the-top reviews to share on STEM Talk. If yours makes the cut and you hear it on the show, send us a note at stemtalk@ihmc.us to claim your very own STEM Talk t-shirt. Proof the committee does in fact exist. Today, our winning review comes from a listener who goes by the name Kiana. The review is titled, My Favorite Podcast, and I Really Want a T-shirt. It reads, Dawn and Ken introduce listeners to such a range of scientists and studies and remind me why I need to always be in pursuit of knowledge. As a young woman in STEM and practicing bootleg science, it's inspiring to hear all of the detail of the scientists' lives and their findings. It is frustrating when I hear facts and figures without a reliable source. But on this podcast, I hear information straight from the horse's mouth. We have to get more horses on the show. I love how the podcast production allows for the speakers to continue on their subjects, no matter how dense. Thank you for making information available to the public. With aloha from Hawaii, Kiana likes you. Well, thank you, Kiana, for such a wonderful review. And thanks to all our other listeners who have helped STEM Talk become such a great success. Okay, and now on to today's interview with Dr. Mari Dezawa.
STEM Talk. STEM Talk.
STEM Talk. STEM Talk.
Well, Mari, welcome to STEM Talk. I'm so pleased you're here with us today. And Mari, you were born in Japan, and your family moved to the United States when you were only 8 months old. I understand that sometimes it can be confusing when somebody asks you where your hometown is. Could you talk about that a little bit?
Okay, thank you. My father, who passed away about 10 years ago, was a vision physiologist of invertebrates. And because he worked at Yale University and University of Munich in Germany, I spent most of my childhood in U.S. and Germany. And my father moved to New Haven, Connecticut when I was a little baby. And we moved to Munich when I was around 4 years old. We went back to Japan when I was in the lower grades of elementary school, but again, we returned to New Haven, Connecticut when I was in upper grades of elementary school and lived there for until the middle of junior high school. My father returned to Japan when I was 14 years old, and since then, we settled in Japan. So when people ask me, where is your hometown? I'm a little bit confused about where to answer. But to be honest, I will say it's United States.
I understand. I also moved around a lot as a child. For some children, it's difficult to move around a lot, and for others, it's not a problem at all. How did you find moving?
Well, we were, just enjoying the life abroad. And it was not difficult for me as far as I remember. So rather, I remember having a little bit hard time to integrate into Japanese society when I returned to Japan and attended the Japanese high school.
Yes, I can imagine it was an adjustment for you. You mentioned your father was a researcher, and so I imagine you've always had an interest in science. But was there a particular moment in your growing up years when you really turned the corner, when you really felt passionate about science?
Yeah. The science was very much a part of my life. And my father's electron microscope picture of insect compound eyes had been hanged in the wall of a dining room instead of oil painting. So in my childhood, my father often took me to his lab on weekends, and I enjoyed microscopic observations and other things. So I loved the smell and the atmosphere of the lab. But when I was in high school, I temporarily dreamed of becoming a lawyer. But the problem was, when I returned to Japan, my score in reading and writing Japanese was really low and devastated. So I found it is hard to keep up with the classes and exams in Japanese. I gave up and changed my policy to go to medical school.
Good choice. In the 80s, you attended the Chiba University School of Medicine, where you graduated in 1989. What prompted you to attend medical school?
Well, maybe I wanted to become a medical doctor to help people.
That makes perfect sense.
Yeah.
I think it's a common motivation and certainly a good one. After earning your medical degree, you attended Chiba University Graduate School of Medicine for your PhD. Initially, you were a cardiology resident before deciding to focus on clinical research and become a stem cell researcher. So what led to that decision?
Well, while studying medicine, I felt that I was more comfortable with logical or basic research than clinical medicine. That was one of the reasons why I decided to become a researcher. The other reason was that the organ transplant that has just started in Japan at that time, I had some kind of vague feeling that there must be a better medical treatment out there, given the various issues with transplantation medicine like immune rejection or shortage of donor organs and leaving the donor family in sorrow and making difficult decisions. At that time, I had the vague idea that it might be possible to use some own cells to cure oneself or to use allogenic cells to provide the treatment that will overcome immune rejection in some way or something like that.
That makes perfect sense, and you've since become known for your stem cell research and investigations into regeneration and functional recovery of nerve and skeletal muscle cells, among others. How and when did you first become interested in stem cell research itself?
Well, when I started my research in the early 1990s, the term regenerative medicine didn't exist, and the concept of stem cell was not that common either. But having an experience as a doctor, I wanted to conduct basic research that will be useful in clinical medicine. My graduate student supervisor, Professor Nagano, who passed away in 2019, was an excellent electron microscopy scholar, and his main subject was a reproductive system. Usually when you join a lab, you are given a research subject, but the Professor Nagano told me to decide everything by myself. I was a little bit confused at first, not working what to do, And after studying a little, I came across a very old book by Ramon Cajal, a great neuroanatomist in the early 1900s. And in it, Cajal sketches describing how non-regenerative central nervous system change and started to regrow when regenerative peripheral nervous segment is transplanted. I was attracted to this phenomenon and began to study the mechanism underlying the nerve regeneration and central nervous system.
So that makes good sense, and I can see the motivation there. Although most of our listeners are well aware of stem cells, and they've certainly been in the media a lot in the recent years, could you give our listeners a brief overview of stem cells and their significance?
So every tissue has its specific stem cells, which are called somatic stem cells or tissue stem cells, or sometimes progenitors. For example, we have hematopoietic stem cells in the bone marrow that continuously generate hematopoietic cells, such as red blood cells and white blood cells, but not liver cells or neural cells. We also know that a small number of neural stem cells exist in the brain that can differentiate into neural and glial cells, but they do not differentiate into heart, muscles, or liver cells. So in this manner, the differentiation of tissue stem cells is confined to tissue component and not to other lineages. So they're a kind of local maintenance company. The Muse cells I found are a kind of general maintenance company in our body. Their reserve is a bone marrow, and they're constantly mobilized into the bloodstream, circulate throughout our body, and supply to every organ and repair the tissue at the microscopic level every day by a unique cell replacement mechanism.
You remained at Chiba University as a research associate once you earned your PhD in 1995, and then you took a position as an associate professor at Yokohama City University Graduate School of Medicine, before moving to Kyoto University Graduate School of Medicine as an associate professor in 2003. At Kyoto, you developed methods to induce bone marrow stromal cells to generate neural and skeletal muscle cells. These cells play a crucial role in tissue repair and regeneration and are widely studied for their potential as therapeutic agents. Can you give our listeners a quick primer on the crucial role these cells play in tissue repair?
Yeah, that's my old version of study. I shifted my research subject from neural regeneration to mesenchymal stem cells around 2000 because I felt limited in only working in a small area. Because MSC started to be focused on due to their ability to differentiate into osteocytes, chondrocytes, and adipocytes. So there were a lot of expectation that MSCs might differentiate into various kinds of cells, and if so, I could find a way to treat the neurodegenerative diseases. So I tried to generate neurons, glial cells, and skeletal muscle cells from MSCs, namely mesenchymal stem cells, by the combination of a gene introduction of differentiation-related genes and cocktails of cytokine stimulations. It did work. I published several papers. But I had firm feeling that the cells differentiating by crossing an oligo-lineage boundary between mesodermal, where MSC belonged, and other lineages where neurons, glial cells, and skeletal muscle cells belong, might be a small percent of the total MSCs. And also many of the MSC researchers had the same feeling as I did. But no one had clues about approaching the genuine pluripotent-like cells in MSC population. So in my bottom of the heart set, I still had not come close to something more essential.
Very nice explanation. Later in 2003, a member of your technical staff at Kyoto asked you whether she should discard some cultured adult rat and human bone marrow mesenchymal stem cells, or as you mentioned earlier, MSCs. She asked you this because she had noticed several strange cell clusters. Can you share what happened next and how this request set you on your path to the discovery of multilineage differentiating stress-enduring cells, more commonly known as Muse cells?
Okay, yeah. The strange clusters were generated spontaneously from rat and human bone marrow MSCs. My technicians said whether she can discard the MSC because the strange clusters were formed spontaneously and our experiment may not work well. So I watched the clusters under a microscope and saw hair-like structures and pigment-like structures in the clusters. And it reminded me of the pluripotent clusters formed from embryonic stem cells, so-called ES cells. So I told my technicians not to discard these cells, but keep them, and then let me know when she found another cluster. Well, I kept on watching them and found several important things. One is that the clusters contain mesodermal, endodermal, and ectodermal lineage cells, suggesting that they're like a pluripotent state. And another is that unlike glial cells, that cluster did not show exponential proliferation. Now, this suggests that the pluripotent-like but non-tumorigenic cells were contained in adult bone marrow cells in rat and human. And if I can find a way to identify those cells, that would be beneficial for treating many kinds of diseases. So I talked about this to my drinking buddies. I had several groups and drinking buddies in Kyoto. We sometimes get together to have fun and sake and wines. And we decided to start the collaboration on defining what cells in MSCs are non-tumorigenic, pluripotent-like, so-called, to pluripotent hunting in MSCs. So we did everything we could do for totally 4 years. And we even dedicated the New Year holidays for experiment, but it didn't work. So then I was fortunate to encounter Muse cells due to my silly mistake. If I were a perfect person, I would never encounter the Muse cells. If I hadn't been invited to the drink party from my buddies, I would never encounter Muse cells either.
That's really interesting. Actually, I think you've probably piqued the interest of our listeners, and I think they might be fascinated to hear in more detail how you described this mistake and how the discovery came about as a consequence of what you have labeled as a mistake.
Well, this is a very funny story. Along with pluripotent cell hunting, we were doing, several other projects in parallel. And one day in hot summer afternoon in 2007, I was doing some simple work, subculturing skeletal muscle cell induced from mesenchyme stem cells, which I told you before. And then skeletal muscle cells, this is a point that contain 3 kinds of cells, namely mature muscle fibers, premature myoblasts, and muscle stem cells. The work was simple, and, I just add the trypsin enzyme, detach the cells, and collect it, and dilute twice, and plate them back to the culture dish. Now, meantime, I had the phone call from my collaborator, one of my, drinking buddies, and he said, "Hey, Marie, what you're doing?" And I said, "I'm doing culture." And he said, "Oh, you're stupid doing an experiment on a damn hot evening like this, so get that over and come with us." I quickly finished subculture and rushed to the drink party and enjoyed that. And the next morning, I checked the cells that were subcultured the day before, and I was shocked to see that it was all gone overnight, disappeared. I couldn't understand what had happened. I was frozen, and I started to recall what I had done the previous night. And I noticed that I had maybe mistaken the culture medium for trypsin enzyme solution because the color was the same. On the other words, the cells were incubated under extreme stress. That's why all of the cells were died. But I had the unique habit. When I make a mistake, I spend about 30 minutes fully enjoying looking at the same, the sample, and then I discard it every time. This is my habit, even now. So from the perspective of nature, I had to believe that, everything is equally a natural phenomenon, even if we think it's failure. So I did the same thing at the time. When I found that, the very small number of the cells were alive, I was excited that amazing to see that cells could survive over 16 hours while I was drinking, the wines and sake. And then those cells had tolerated a very harsh environment, no nutrition, only with enzymes. And the next moment, anyway, the old cells were gone, but instead, very small number tolerated. I collected those cells and found that those stem cells had survived. This is scientifically reasonable because stem cells are usually stress tolerant than mature cells. So I quickly called the guy who called me the day before, and I told him what had happened. Only the muscle stem cell remained. This is what I have found. And he said, "Oh, that will be a lucky turning point, Mary, and maybe we can apply that to the pluripotent stem cell hunting in MSCs." So then we seriously reproduced my mistake, but without drinking this time, and then finally succeeded in finding Muse cells among the MSCs. That was in 2007. But the first announcement of a Muse cell paper was in 2010. The reason why it took 3 more years, that is because we have to struggle some more to find a robust marker that would allow others to reproduce Muse cells. It was a physically demanding work, and we finally found that the cell surface marker SSES3, a commonly expressed, the prepotent surface marker, can be used to identify and select Muse cells. So the guy who called me that has an excellent idea to include stress enduring into the full name of the prepotent stem cells we found. And after discussion, we named the multilineage differentiating stress enduring cells.
What a wonderful story. And one way to interpret the story is a tale of good fortune. Another way to interpret it is good fortune coupled with a prepared mind. And so you were ready at the time to receive the good fortune. In 2008, you became a professor and chair of the Department of Stem Cell Biology and Histology at Tohoku University Graduate School of Medicine. And as you mentioned earlier, in 2010, you published a paper on the results of your investigations into Muse cells. This is really a significant paper in my view, and it set the tone for much of your future research. And you started to talk a little bit about this, but could you further explain for our listeners some of the characteristics and advantages of Muse cells?
So Muse cells were originally considered very convenient prepotent stem cells with a tumorigenic property. But as research has progressed over the past 15 years, the interpretation of the Muse cells have changed slightly. The biggest difference is that there are special in vivo stem cells that resemble immune cells with an aspect of a prepotent stem cells. In other words, the Muse cells were macrophage-like and prepotent-like endogenous reparative stem cells. So that the main reserve of a Muse cell is in the bone marrow. They're mobilized at the constant rate into the peripheral blood. They circulate the body and are supplied to every organ. In our body, we have damaged cells and dying cells, namely apoptotic cells at the microscopic level every day. So those cells produce a universal damage signal, namely sphingosine monophosphate. The Muse cell sharply sends sphingosine and like macrophages, they capture damaged cells and apoptotic cells and eat them, like phagocytose. Then, Muse cells receive the recycled factors necessary for differentiation that were active in phagocytose cells and quickly differentiate into the same cell type and replace damaged cells with apoptotic cells. And in this manner, they repair the tissue. So, since Muse cells are pluripotent, they can differentiate into various kinds of cells, including vascular cells, that is necessary for nutrition of the tissues. When we utilize Muse cells in clinical treatment, there are mainly 4 major advantages. First, Muse cells do not require donor selection. Due to specific immune privilege, we can directly use HLA mismatched donor Muse cells for intravenous injection. Immunosuppressants are not necessary. The clinical trials conducted by donor Muse cells without HLA matching test or immunosuppressant show that donor Muse cells were not rejected and recovered functions were maintained for several years. Up to now, it's 7 years since we had done the first clinical trial, but still there are no signs of immune rejection up to now. Now, second, intravenous injection is the most efficient administration method. It does not require surgical operation to deliver Muse cells to the target organ. Now, differentiation, induction, or gene introduction are all unnecessary. Now, since Muse cells survive and remain integrated in a recipient tissue, their bystander effect like antifibrosis effect, neovascularization, immunomodulation, and the tissue protections are all long-lasting. And therefore, Muse cell treatment can be done in a simple way, namely intravenous injection of HLA mismatched donor Muse cells without immunosuppressant, gene manipulation, or differentiation induction prior to treatment.
That's an exciting list of characteristics and advantages.
STEM Talk is an educational service of the Florida Institute for Human and Machine Cognition, a not-for-profit research organization investigating a broad range of topics aimed at understanding and extending human cognition, locomotion, health span, resilience, and performance.
As you mentioned, one of the key characteristics is that Muse cells can essentially repair all organs, and they also quickly repair themselves. And I think this is part of the fascinating stress resistance that you mentioned earlier. Could you explain briefly, I know you touched on it earlier, but could you explain briefly the significance of this sort of self-repair and stress resistance?
Yes. As I mentioned earlier, they're stress tolerant and can survive in harsh environments such as under low or no nutrition, even and enzymatic solutions for longer than overnight. Now, this is partly due to the high expression of stress tolerant related factors such as serpins and 14-3 proteins. What is more significant is that even if their DNA is damaged or cut by UV or X-ray radiation or by exposure to toxic substances such as hydrogen peroxide, they can quickly finish repairing damaged DNA within 6 hours, while other kind of stem cell needs more than 48 hours. So Muse cells not only repair the whole body, as a general maintenance company, but they can also repair themselves for their own missions.
Very interesting. I'm curious, Mari, how are Muse cells collected and formulated? How do you collect them?
Okay, the Muse cells can collect from various tissues because they're found in the bone marrow, peripheral blood, and connective tissues of every organ. But the practical sources will be umbilical cord, adipose tissue, and bone marrow. Now, umbilical cord Muse cells will be one of the best options for allogenic treatment, and adipose and bone marrow Muse cells for autologous treatment. In those tissues, Muse cells are always contained as SSES3 positive cell. The point is that Muse cells are expandable to the clinical scale, and their doubling time is comparable to that of fibroblast. So, namely, 1 day or 1.3 days per one cell division. Is almost same as that of fibroblasts. So there are several methods for formulation. Either way, we first establish MSCs from the umbilical cord, adipose tissue, or bone marrow, and then increase the percent of the Muse cells from, originally they were containing those MSCs from 1 to several percent, like, 3% or 4% or whatever. And then we will increase the percent of the Muse cells or the purified Muse cells from MSCs. One of the methods to expose MSCs to several stresses in step-by-step manner and elevate the percent of the Muse cells from several percent over 70%. So Muse cells survive because of the stress tolerance, but the cells other than Muse cells in MSC will die because of the stresses. Another method is purify Muse cells by using their specific surface marker, SSES3. And in this case, we need a magnetic activated cell sorting device, such like MAX, so-called MAX.
Since the 2010 paper that we discussed earlier, that paper detailed your early investigations and discovery of Muse cells. You've published the results of several follow-up studies. In 2018, you published a paper that looked at the use of Muse cells in the treatment of acute myocardial infarction. And this, as everyone knows, is a common cause of morbidity and mortality worldwide. Please discuss this paper and its primary findings.
Okay. Now, first, the intravenous injection was shown to be very efficient approach for treating diseases because Muse cells sharply sense sphingosine monophosphate, a universal damage signal common to all organs. They pass through the lung. This is a very key point. They pass through the lung and are recruited to the damaged tissue accurately, deeply, and extensively. This is one of the significant difference to mesenchymal stem cells because mesenchymal stem cells are mostly trapped in the lung after intravenous injection and disappear from the body within several weeks. So MSCs deliver only temporal bystander effect and do not contribute to structural and functional restoration of the damaged tissues, unlike Muse cells. Now, the second point is that the homing rate of the Muse cells intravenously injected to target organ was found to be around 15% of the total. However, MSCs were under detection level in the targeted organ. And thirdly, the intravenously injected Muse cells finished differentiation into physiologically active cardiomyocyte by 2 weeks. It was so quick. And fourth, the therapeutic outcome did not differ significantly between autologous and allogenic Muse cells. This is also a very big point. And it was amazing for us to find that allogenic Muse cells survived in the recipient heart tissue as a cardiomyocyte for up to 6 months without immunosuppressant. So based on G's result, the first-in-human clinical trial was conducted by intravenous injection of only 15 million donor Muse cells without HLA matching test or immunosuppressant, showing significant left ventricle ejection fraction recovery from 40 to around 53 by 3 months. And 15 million cells are only, 10 to 15 times lesser clinically used in MSCs.
Really interesting. Stem cells undergo cytokine-driven differentiation. This process often takes a while, longer than several weeks to complete. However, you uncovered a simple mechanism that could substantially shorten this time frame. You reported on this in a 2022 paper titled Phagocytosing Differentiated Cell Fragments is a Novel Mechanism for Controlling Somatic Stem Cell Differentiation Within a Short Time Frame. Can you talk about this paper and the potential implications of its findings?
So whenever I give a lecture, I always got a question, okay, so prepotent ES cells or iPS cells will differentiate randomly if, let them go for differentiation. Why can Muse cells become correct cells compatible with a tissue after homing there? And we felt that was, very major scientific question, but we could not come up to the solution very quickly. However, we had some, come across a hint a long time ago. When we first discovered Muse cells, a collaborator told me that the Muse cells were doing something like phagocytosis. This is very, weird, but very interesting report. At the same time, we found that they were expressing some of the macrophage monocyte-related genes or markers, along with pluripotency and stemness. So one day I imagined that, so Muse cells were phagocytosing, receiving differentiation signal from there and differentiating into the target cell type. So we tested it, and it turned out to be a hit. And Muse cells have many characteristics similar to monocytes and macrophages. This is really true. So both reside in a bone marrow. The Muse cells and monocyte macrophage and lineage cells both reside in the bone marrow. And the size and shapes are almost the same. And both are recruited to damaged tissue by sensing sphingosine monophosphate. And both express receptors for phagocytosis. So they are very close to each other. But the Muse cells have pluripotency, but monocyte macrophages, they do not. The system is very reasonable. When tissue is damaged, Muse cells and monocyte macrophages are recruited to the damaged site, and the macrophages are professional phagocytes so that they clean up the tissue. But Muse cells also clean up, but their primary mission is repair the tissue by phagocytosis-dependent differentiation. Importantly, the phagocytosis-dependent differentiation progress in days, very quickly.
I find the similarity to macrophages particularly interesting and actually somewhat surprising. So that's very, very interesting, Mari. At IHMC, as you probably know, we work extensively with the Department of Defense and the various branches of the military. So I'm curious about your thoughts on the potential applications of Muse cells for military applications. Several pop into my mind already, so I'd love to hear your thoughts on this.
The first Muse cells can be frozen and stored for long periods so that they can be used in areas with a poor access to medical care, such as combat zones, mountain areas, deserts, remote islands, and even in outer space. Moreover, they can be administered intravenously to repair various organs at the same time without need for complex procedures such as HLA matching tests or surgical treatment or whatever. So for military use, Muse cells can be used for example, mild traumatic brain injury, brain hemorrhage, or PTSD, or even dementia by nasal drops. This is very, easy to treat with. And, for example, another will be cytokine storm induced by infections. And thirdly, the injury and wounds and in joint, cartilage, muscles, and skins. And treatment and pre-treatment for lethal dose radiation and also including anti-cancer.
What about exosomes? Can you explain for our listeners what exosomes are, and in the context of Muse cells, how might they be applied?
For military use of a Muse exosome is very vital, and because exosomes can be preserved even in crude ways. They can be stored at room temperature and more stress resistant than Muse cells itself. So Muse exosomes can be used for combat zones and pre-treatment for mild TBI, traumatic brain injury, and brain hemorrhage, PTSD, and dementia by nasal spray, and pre-treatment for injury wound and by spray with pre-treatment for sports medicine, joints, and cartilage is done by local injection. And in terms of easy of storage, the Muse cell exosomes are somewhat superior to Muse cells themselves.
You mentioned just now the nasal administration of stem cells, and you reported on this in a paper titled Nose-to-Brain Delivery of Human Cells Enhances Structural and Functional Recovery in the Murine Ischemic Stroke Model. Can you talk about how intranasal administration of Muse cells might be a viable approach to improving functional recovery after somebody experiences an ischemic stroke?
The roof of the nasal cavity is supplied with sensory neurons that sense smell. And when the molecules bind to the nerves, they react excited, and this excitement passes through the tiny holes and ethmoid bones at the base of the skull. It enters into the brain and transmitted to the olfactory bulb. So therefore, the nasal cavity is connected to the brain at the very microscopic level. However, not everything can pass through. The Muse cells can pass through it easily when human Muse cells were administered to the nasal cavities of model mice with cerebral stroke or TBI or Alzheimer's disease. They can quickly migrate into the brain and produce the same therapeutic effect as when administered intravenously.
Very, very interesting. I understand that you have a paper that's in development, preparing for submission, or perhaps may have already been submitted, on the use of Muse cells in the treatment of traumatic brain injury and perhaps even dementia. Could you talk about that a little?
So TBI, so-called traumatic brain injury, can be treated both by intravenous injection and nasal administration. In Alzheimer's disease model, intravenous injection and intranasal did not differ in their therapeutic outcomes. This was really also amazing for us. From these data, we can foresee several strategies. If the target organ is confined to the brain, for example, the first choice will be nasal drop because we can do that quickly, even by ourselves or oneself at any place, and it can be repeated for multiple doses for preventing worsening and to promote repair quickly. For dementia, as it progresses chronically, multiple doses at certain intervals are required. So the nasal drop can be done even at home. However, if multiple organs are damaged, such like in the case of TBI, like bone fractures or muscle diseases or skin, intravenous injection will be the first choice.
Our listeners always wonder about models and about how well the model replicates the system of interest. In the case of, say, Alzheimer's models, they tend not to be very good, not very much like humans. So I know you've had work looking at human trials. Could you talk a little bit about where that stands and where you hope to go with human trials?
Well, we're also surprised that, in clinical trials. It is too good to be true. And then initially I thought or imagined. But the doctors that have actually experienced the effect of the clinical trials, and they said with a shine in their eyes that it's more effective than any other treatment they've experienced. So I think it's probably true. So, however, there are some problems. Is that the important to maintain the percent of the Muse cells over 70%. This is a gold standard for Muse cells that bring us and yield the outcomes almost comparable to 100% pure Muse cells. So, if this is not done properly, for example, if the content of a Muse cell is less than 50% or even 30%, the therapeutic effect will be greatly reduced, and the result will not be that much. So this is a point that we have to always be careful of.
Thank you for that. And a little bit later today, you'll be touring our new HealthSpan Resilience and Performance Research Complex here at IHMC. As you know, there is finally a burgeoning research effort aimed at prolonging human health span. What have you learned about MSCs and Muse cells and their possible use for health span optimization?
Well, aging is based on cellular changes, including terminal attrition and DNA damage and mitochondrial dysfunction, and it occurs throughout the body, so the entire body is targeted in aging. In that respect, Muse cells, particularly younger tissue-derived Muse cells such as umbilical cord Muse cells, will be a good fit because they are slightly higher activity than adult-derived Muse cells. And Muse cells and MSCs are fundamentally different, as I told. The Muse cells can structurally and functionally repair the various tissues by cell replacement, but Muse cells deliver temporal bystander effect, and they're mainly trapped in the lung after intravenous injection. Indeed, the superiority of Muse cells over MSCs and therapeutic effect was shown in the brain, skin, spinal cord, heart, lung, liver, kidney, joint cartilage, and even in genital organs in preclinical studies. And in clinical trials, Muse cells were shown to be therapeutic in acute myocardial infarction, subacute stroke, epidermal bullosa, ALS, and neonatal hypoxic ischemic encephalopathy, and even in cervical spinal cord injury. So therefore, Muse cells might be a good use for mesenchymal stem cells. Enriching and purifying Muse cells will enhance the therapeutic effect of mesenchymal stem cells, leading to further expansion of the use of MSCs, even for health span.
That sounds very promising. Well, Mari, we really appreciate you taking the time to visit us here in Pensacola, and we do understand how busy you are. In addition to your work at the university, you've also joined forces with Muse Cell Innovations, a company that is working to revolutionize regenerative medicine and longevity through advances in cellular science. The company specializes in advancing the potential of Muse cells in the hopes of creating products that they believe will empower people to live longer, healthier lives. In terms of full disclosure, both you and I serve on the scientific advisory board of Muse Cell Innovations, and that is how we got to know each other. Can you talk a little bit about your involvement with Muse Cell Innovations and how it all came about?
Ken, I'm very honored to work with you. The Mitsubishi had doing the Muse cell business since 2015, but it withdrew from the business in 2023. It wasn't due to technical problem with Muse cells or a problem with therapeutic effect, but was a decision in the company's management. And regenerative medicine takes a long-term approaching, and it's not the field that generate immediate profits. So I was looking for a partner company and started to explore overseas possibilities. This is because, Japan's market is not that big in the global medical market, and that accounting only for 4 to 5% of the total. And rather than fighting in Japan, Mitsubishi's withdrawal was a turning point at a rather good chance for Muse cells. This is what I have told. And 2024, I received the offers from several companies from the U.S. and other countries. But I felt that I could achieve my goal with members of Muse Cell Innovations. So I decided to assign the patent rights to Muse Cell Innovation and entrust the future of the Muse cells to them.
Well, it is an exciting development. Mari, our conversation today has certainly been a lot of fun. And as we wrap it up, I'm reminded of an interview you gave a few years ago where you were asked how you balanced a life and a career. This is a topic that seems to come up more and more these days. You said having a hobby helps. Okay, but you never said what the hobby was. Can you share that with our listeners?
Okay. Well, I can relax in nature. I love nature, so I like hiking and sometimes challenge myself with mountain climbing, but not that very high, low mountains. I enjoy that. I also enjoy drawing pictures because my mother is artist, and I also play piano. And at my time in medical school, I also had the violin to play in a part of orchestra. But not in recent day because I have no time to enjoy my hobbies, but I hope to do so in some days.
Well, that sounds wonderful. And as our listeners have already no doubt guessed, if I tell them we overlap on one of those hobbies, I'm sure they have already guessed that it's hiking in mountains.
Yes.
So that is my favorite as well. So thank you very much for joining us here on STEM Talk.
Thank you, Ken.
STEM Talk. STEM Talk.
STEM Talk. STEM Talk.
What was so wonderful about our conversation with Mari is that her research truly exemplifies the remarkable potential of regenerative medicine. The therapeutic possibilities of Muse cells are extensive, and their importance is such that they deserve far more attention in research. Like many breakthroughs in science, their discovery had an element of serendipity. And as we discussed with Mari, she and her team noticed that a rare subset of stem cells survived stress conditions that killed most others, an observation that led to the identification of Muse cells. This element of serendipity in science is relatively pervasive and really just requires a prepared mind to see the serendipitous result. Very interesting. So stay tuned. We're likely to be hearing much more about this exciting area in the years ahead. This is Ken Ford signing off. Goodbye until we meet again on STEM Talk.
Thank you for listening to STEM Talk. We want this podcast to be discovered by others, so please take a minute to go to iTunes to rate the podcast and perhaps even write a review. More information about this and other episodes can be found at our website, stemtalk.us. There, you can also find more information about the guests we interview.